Wait, what's going on? What is that? Was that there? Okay, I'm on a stage. Wait, what's going on? What is that? Was that there? Oh my god, what is it? Why did it become a horror game? Why did it become a horror game? What the fuck is that shit?
Yeah, what do you need me to do?
I don't know.
Okay, I see things I can move back and forth. I see a crown, a sword with a crown,
Hey.
3 happy people dancing, a castle.
Let's get started.
Castle.
What about the castle?
What can you do with... Oh, did that thing just move? What? Holy sh...
Recording in progress.
What can you do with the castle?
Talk to me. Is there things...
Okay. Before we begin today,
Filled with life. Filled with life.
Life.
Yeah.
I know everyone's excited. We're almost done this semester. I am very excited too. Can't wait. So what? Next week is the last week. Wow, things are moving fast. So Deep Question 5 is going to come out tonight. It'll be released this evening. And then all we have left is the final take-home questions. So we'll get to that in a sec. But I wanted to advertise this. I did send out a message to everyone. And wow, this is super loud today. I sent out a message. I know what you're all thinking. It's exams. Nobody's really thinking about what they're going to do in their next year or maybe in their years after. But I wanted to advertise this honors research opportunity. I'm actually looking for 2 students to start in the fall, maybe in the winter. I don't know. We'll see how the candidates are that apply. Not in a huge rush, but I like to take on 2 students per year. I like to cast a wide net as possible. I realize that many of you are in your second year, so honors is not even really an opportunity for you at this point. You have to be, I think, in your final year, and there's a certain number of credits. You'll need to check the criteria on the message that I sent out. But I just wanted to give you a quick, because I sent out the email, it didn't really explain much anything about the opportunity. So why do honors in the first place? I think every year there are many, many more people that want to do an honors project than positions that are available. Honors will help you in your career. Whether your career is going to be, you plan to, next step is going to be getting a job in industry right from the undergrad, or it's med school, grad school, dental school, law school, whatever. I've had them all come through my lab. It helps you separate you from the rest of these, from the rest of your peers. Having that research experience is, especially for med school, but anyways, it really helps distinguish you. And it's not just for the career opportunities. I would say the other reason why it's really important is research is a total other level. It's a total different thing. It's very different from classes. It's a fantastic experience, discovering new things that no one else on the planet knows. It is really exciting. So the opportunity, I have 2 honors research projects in my lab each year. If you don't fit the criteria this year, you can always maybe reach out to me around this time the next year. But anyways, I got a pretty cool crew. Unfortunately, we haven't updated our website with the current group. Some of them are been here for a while, and we have other new ones, and possibly you could be added to this. We update the web page every summer. Really great group, very collaborative, interactive group. So a lot of different mentors. And as a student, you'll learn things and you will actually get to mentor other people. So we teach each other. What are the projects about that we're studying? We're researching essentially plant bioengineering for pharmaceutical production. So we're looking at how plants produce pharmaceutical metabolites. Right now, we're studying gene regulatory networks in, believe it or not, this is soybean here with the roots growing out of the leaves. This is bioengineered soybean roots and cannabis plants. We're essentially studying gene regulatory networks. That's a fancy way of saying transcription factors and their targets and how these networks actually regulate the production of valuable molecules that we want to learn how to produce in higher quantities for the pharmaceutical industry. Okay, so if that sounds to be of interest to you, you can always send me an email. If you're really interested in the honors projects, check out the email about the criteria. So there is a certain set of criteria needed to be in the program. Yeah. Honors thesis. Honors thesis, that's for Bio 4000, the 8-credit one, the research thesis. Okay. All right. Okay, moving on. All right, so today we're going to do an activity, as usual for Tuesdays. Next week, last activity. So tonight, deep question gets released, the last deep question. Next week, it's actually, if you check the calendar, it's scheduled for Thursday, the 6th. That's whenever the final take-home questions are supposed to be released. I'm actually going to try to get it to you earlier than that, because I think many of you, like myself, would like to take the weekend off and not worry about it. I'll figure if I can get it out to you by Tuesday. I'm going to try to get it out, released by next Tuesday. It'll give you more time. Next Thursday, we'll have the class to work on it. Okay, so you can come in and it's just like the deep questions. You can work together. Careful about following down the rabbit hole the wrong answer. It'll be very similar to what you're already used to. I'm not sure if I'm going to give you more or less than usual, but considering the time, because they're due by the end of class, which is by the end of the course, which is, I think on the 10th, I'm going to give you one extra day. So I'll give you to the 11th to get them done. Okay, so any questions? Yeah. No, I think it, if I release it next Tuesday, which I believe is it'll be like 1 week. Yeah. Yeah, 1 week. And again, like should take you like an hour, but if, these are finals, so you might want to put some more into it. It's nothing, you don't find anything on the internet. It's everything came from the class. Okay. So, it's typical stuff. Right. Yeah. You'll have about a week to do them. Last year they released them on the Thursday and had them due the following Tuesday. So it was less than a week. I'm going to try to give you a full week. Okay. I'll try to get them done before the weekend, just so you don't have to worry about it. Enjoy your weekend, relax. Get the course done. Okay, learning objectives. I'm not going to go over these. You're already aware of them. RNAi, gene silencing, X-linked things. They had a beautiful activity on X-linked things last year. Oh, I cut it out this year. But anyways, RNAi is cool stuff too. So, before we get into the RNAi, I just want to review from last class because I guess last year there was a lot of difficulty on the GWAS, understanding the GWAS, and we went over it. For those of you that actually came to last Thursday, we did talk about the GWAS, and we talked about how just because you find a SNP that's highly correlated with a particular phenotype doesn't mean it's causative, doesn't mean that it causes that phenotype. There are a lot of different possibilities, and they're shown here. So sometimes maybe you can identify a SNP like these guys here. They're not really affecting the protein production or function or anything nearby. What they're doing is they're just segregating with maybe other genetic differences that we're not following on the SNP chip. And we would have to then go into that genomic region and try to figure out what differences might be causing a phenotype of interest. Okay, so anyways, the SNPs can be in non-coding intergenic regions. They can actually be in coding regions. It does happen, and they could cause a difference in the amino acid sequence, but maybe not. Could be in regulatory sequences. They could be completely unassociated with the gene, which just makes it much, much more difficult to figure out what on earth is going on here. But anyways, usually the case is that whatever the SNP that you're finding that's correlating with your gene of interest, it's going to be something nearby that the SNP is affecting somehow. Okay? Yeah. Yeah. Yeah, that's a really good point. So the question is about if the SNP is really, really far away. Actually, sometimes they could be like tens of kbs away, right? And so, yeah, and so the SNP would only be following that causative. It would only, how do I say this? It would only be meaningful and found in our SNP chip array if it was following because sometimes it doesn't have to be close. Actually, sometimes it can be 10 to 20 kb away, and it's just there's sometimes recombination hotspots are the opposite, where recombination doesn't happen very often. And if you think about it, so in other words, even though it's far away, it can follow the gene of interest around. I try to, so this is just a made up scenario, but imagine the SNP, it actually encodes a regulatory element like an enhancer. And we know enhancers generally can be very, very far away, right? And so if that enhancer has a phenotypic consequence, it is activating a gene of interest, and we are following different alleles of the enhancer. If there is recombination, that essentially that whatever the A sequence in the enhancer or the C or the G or whatever it was that we're following, its association is lost, right? And so we would only find it if it was associating with. So we would find the A, we wouldn't find the other ones, and then it's up to you. So do you know that 20 kb away there's an enhancer that it's really hard to find, right? It just becomes very hard to find. But that whole finding part is another ball of wax that we don't really go into. But yeah, if there is combination in between. You can imagine anytime it is inherited, it that enhancer is going to follow, it's going to have then the effect, right? Even though the effect is on the gene, which is far away. So that's the idea, right? Is that you may look at the genome and be like, well, what is that? Like, there's no coding sequence. There's no even coding sequence nearby. What on earth? And the next neighboring gene might be 20 kb away. Or, to make matters worse, we won't. There's even worse, like complicated scenarios where enhancers on one chromosome are actually acting on different chromosomes, and that just like boggles my mind. But you wouldn't find it if you were looking on the same chromosome. So it can be sometimes we just can't figure out what the SNP is doing. We just know that it's causal somehow or linked somehow. So that whole linked versus causal, you have to dissect. Okay, I'm going to try not to get too off on a tangent, but we did discuss last class though how you do that, right? And how you can check if a SNP is the causative agent or not. You can go in there and you can mutate to lose the SNP. You can cause mutations that gain the SNP where the SNP wasn't present before, just to see if they actually cause the phenotype or not. Yeah, okay. So, what this whole GWAS thing is like, this example, we're looking for, I guess, dogs of different fur color, dark fur color versus light fur color. And then we have all these different SNPs. And in this particular instance, this SNP completely associates with the fur color. Okay, so whenever you have your thiamine at this particular position, you have a black fur color, and whenever you have an adenine, you have a white fur color. So totally correlates. Anytime I have a T, I have a black fur color, and anytime I have an A, I have a white fur color. So remember that we're not just doing 8 individuals here, you're doing generally thousands, right? And so you're this having a very complete association would be strikingly high on your SNP array. You're measuring the amount of times it associates with a phenotype or not. So it'd be very, very strongly associated with the color. And then you have other situations like these other alleles. This one, for example, so we have G, G, G. These guys, they're all Gs, 1C. And then we have 1C, G, G, G. So we found the same thing for the black fur compared to the white fur. So they're completely unassociated. There's no association whatsoever. They would not score highly on your SNP chip result because there's no association in that, and that's what you're looking for. And that's the idea, So you're going along the length of a chromosome, and again, this is one chromosome. We're zoomed in here. Remember, we do this for all chromosomes, but we're zoomed in at this point, chromosome 32, and we're looking at various positions along the chromosome. And so which one of these has the strongest association with the trait, which is, I guess, coat length in this case? Yep. Three, 337. Yes, exactly. It's this one right here. And so see, it has the highest, it's measuring highest on the total number of differences. This SNP chip chart, and it's, I don't really like the units they put here, but it's the log P of the blah, blah, it's the P value. So it's association with the trait of interest. The bigger the bar, the more that this particular SNP along the chromosome, and we're looking at a few of them here, this particular SNP is correlating with the fur color. Or I guess coat length in this example. Which one is least correlated? So which one would you expect it? You just have random, everything you find for one coat length, you're gonna find it for another coat length. Yeah. Right, exactly, the 804, this one here. We're not seeing any correlation. So the SNP is not following it at all. It's not enriched at all for that code length. So that's the idea of the SNP chip. And so now that we found this, we think this SNP, this particular SNP right here, we'd have to, we'd have to zoom in, click on it, whatever you want to say, to find out what allele it is. Is it an A? Is it a C? Is it a T, a G? Whatever. And then we would actually go into the genome, look at the particular sequence, look at neighboring genes, and try to figure out what could possibly be causing this difference in length. This is just a flag drawing our attention to that particular place in the genome. Okay, so that's GWAS. Okay, so during a lab meeting, you're reviewing your GWAS work on short and long-haired dog breeds. Your friend asks you whether this was done across dogs from a variety of breeds. Why would this matter? How might it affect the results? Go ahead, take a few minutes to discuss amongst yourselves, and then we'll take it up. Okay. Anyone want to volunteer? Yes, yes. Oh, yeah.
Long-haired dogs, then when we analyze their SNPs, we might be able to find certain correlations. We don't know the big picture.
Very good answer. You're not exact. So it was one breed enough. Very good. One breed, not enough. I got a boxer. Short hair. That's short. I don't know. Sorry, I think that. It's funny because I've had boxers, they all have short hair, right? I don't know if you know your dog breeds. Very short hair and, cute dogs. Anyways, short hair, so you don't have a lot of variability in the hair length. And if you only focused on one breed, have you ever heard of the founder effect? What is the founder effect? In a nutshell. Do you remember? I'm going to pretend for a second that I don't recognize people from plants because I talked about it in plants. Yeah. Yeah, it's so, yes, this is a physical example, physical isolation example. A part of a population moves to an island and now they're geographically isolated from the rest. So that small group, there's 8 of us and we move to an island and then we populate the island, future generations to come. Okay, a very small representation of the original population. And starts a this is what the new dog breed is, right? They find the traits that they like and okay, we're gonna breed these two. They look very, very similar. We're gonna try to get the same trait again and again and again. They establish a breed. It's the founder effect. And essentially what they're doing is they're reducing genetic variability. But for GWAS, you want to have as much genetic variability as you possibly can, right? So it wouldn't be a good idea. You know, you did your research and you did only do it on one breed. There's got a problem here. So that was a good question from your lab mate. How How might it affect the results? Well, how might it affect the results? You chose one breed, not all the dogs, all the possible variation. Yeah. Hmm. Yeah, you would have variation for everything that the breed varied for. I don't know if that's making sense, but if hair length wasn't highly variable, you would have, how do you look for variation in genetics that causes variable hair length if you don't have variable hair length? It's, yeah, fur length. Okay, so great. Okay, so today we're going to move away from that. We're going to talk about types of RNAs. Who can remember the different types of RNAs that there are? 6 of you. Awesome. Okay, so today we're going to talk about there are various types of RNAs, mRNAs, tRNAs, involved in translation, ribosomal RNAs, so on and so forth. Today we're going to talk about microRNAs and small interfering RNAs, these two guys here. Okay? They're involved in regulating gene expression. MicroRNAs, siRNAs. Look at, they draw them like this, little bump. And these ones are not. So that actually means something. But anyways, so how do these regulate gene expression? Do you remember? Have you seen these before? Did you watch the pre-class videos? Okay, all right. There's some people that watch those videos. Good. Do you remember how did these work? How do these work? Yeah. Okay. Yeah, very good. So you have these affect the transcript. They're gonna do something to they regulate gene expression. You have an mRNA, so a gene, typical gene that's transcribed. It encodes a protein, for example. It doesn't have to, but it encodes, in most cases, it's an mRNA that encodes a protein. And then these microRNAs or siRNAs, they're going to bind. They're complementary, reverse complementary. They're going to bind their target. Okay? Great. These are small RNAs. And only, 24 nucleotides. Evacuate. Only about 20 to 24 nucleotides long. And so we're going to get into the differences between the microRNA and the siRNA, but essentially what they do is like miRNAs. You see that little bump here? This is a creative way to say, well, this is, it's going to bind a target RNA. It's going to be complementary to a target RNA, like an messenger RNA, but it's not going to have perfect sequence complementarity. There's going to be a little bump that's not going to be complementary. I presume that's what they're talking about here. And so that little mismatch is actually it's gonna bind, and it's going to do what to that mRNA? What does it do? Yes. Not in this case, not when there is a mismatch. Whenever there's a perfect match, like is in the case of the siRNA, and sometimes miRNAs are a perfect match. If they're a perfect match, reverse complementary, and all the bases match, all the 22 to 24 bases match, you are correct. There will be enzymes that we're going to go into, risk and so on, that are going to cut the target, right? But in the case when they're not a perfect match, there's one mismatch or two mismatches, what happens? A different process happens, the target does not get cut into bits. Yeah. Yes, it remains bound, and what happens? So the proteins that are holding the mRNA, they remain bound, and I can't cut this. It's not a perfect match. It doesn't line up, and they just remain bound, and they block translation. We had two levels. You said one of them. One of them was to cut the RNA. So your transcript, the stability of that mRNA is dramatically reduced. It's actually degraded. It's going to get chopped up into bits. So no chance of translation at any time. It gets cut up. But on the other hand, whenever there is a mismatch, it can block the translation. So that's two levels. They're both post-transcriptional levels. Happens after transcription is done, but they're different. Okay. So we have our miRNAs and our siRNAs. miRNAs are generally, every year, I made some hard questions on this last. It wasn't purposefully hard, but the class really had a hard time with this last year about miRNAs. And miRNAs are actually, so they'll have a target somewhere in the genome. They have a target, a target gene. It's going to be transcribed. It's going to make an mRNA, and that mRNA is its target. It's going to try to get that mRNA and bind to it. The miRNA itself is actually encoded somewhere else in the genome. Oftentimes, it's not encoded by that same site. It's encoded somewhere else, and it's encoded by inverted repeats. This is what everybody thought was very confusing. It is encoded by inverted repeats. Let me see if I can draw this out here. I'm just going to draw out what in it. So imagine this. I have my strand of DNA, 5' end here. Apologize for and I'm going to say this is like the code here. Oh my gosh. I should have brought my pen. My daughter's 5 years old. She'd be laughing at me right now. Okay, so A. Let's just say that this was the code. And then we have some nucleotides in between. I don't know what they are. I'm just going to put N. Okay, and then I have the other part of the miRNA. And I'm talking in the genome. This is not the mRNA itself. In the genome, and this is T, and so on, 3' end. Whoa, oh my gosh, you can do it. Okay, wow, that looks terrible. This is the 5' end. And then we have the A. I put my arrow facing this way. It doesn't mean it's transcribed this way. What happens is it's transcribed like this, all the way across as one transcript. And now what happens whenever this is transcribed and we have these nucleotides here are complementary to these nucleotides over here? What do you see in your mind happening? Yes. They're going to form a hairpin. They're going to bind to each other. Okay, they're going to make a double-stranded RNA. So this thing that gets transcribed up here, ultimately what it's going to do, I'm going to show you my AAA arrow here. And this NN in between, the NN doesn't bind anything. But now I'm going to draw this other side of the TTT over here. Okay, and these guys are going to base pair like this. And they're going to form a double-stranded RNA. This was the 5' end. This is the 3' end. I'm drawing over stuff. I'm really not, yeah. Okay, so yeah, the RNAs are like, the repeats are facing like this. It's an inverted repeat transcribed all the way through. You have a transcript that's shaped like that, and it base pairs, makes double-stranded RNA. That's all that's caused, that's all you need to trigger this regulatory mechanism, the miRNA or the siRNA mechanism, is to have a double-stranded RNA. A double-stranded RNA is going to be recognized by an enzyme called dicer. Dicer comes in, chops it up into little 20 20 nucleotide, 24 nucleotide bits. And then another enzyme, RISC, comes along, takes those bits, denatures them. And what it does is it, like a little radar, it looks around, it looks for complementarity. It's checking things out, checking for complementarity. As soon as it finds complementarity to a target, finds the target and it cuts it. If it's totally complementarity, it cuts it. If it's lacking total like if there's a mismatch of a base or two, it just remains bound and it's going to block translation. Okay. Okay, so more than half of miRNAs are what we call intragenic. Intragenic, they're within a gene. Only about half, more than half, okay? And the other half are intergenic, so they're between genes. Okay, so I guess we're referring to protein coding genes. So more than half are in the protein coding genes, and they can be, if they're within the protein coding genes, they could be in an exon. Okay, so that's what this loop forming right here is. Like I told you, that's the hairpin inverted repeats forming like this. Okay, so it can be in an exon. Oftentimes it's in an intron. So it's gonna get, this intron will get spliced out. This mRNA gets transcribed. The intron gets spliced out, so the exon gets linked to an exon. And when that intron gets spliced out, we generally think of introns are they're not really useful, right? They get spliced out, and then they get degraded, they get all chewed up. Not in this case. In this case, an miRNA hairpin forms, and guess who's gonna find it? Dicer, and then RISC. So this mRNA is actually gonna make this intron is gonna make an miRNA. It can also be intergenic, so between genes elsewhere in the genome. Remember, the genome is transcribed. We learned in the first lesson that the bits of the genome are transcribed all the time and all over the place. It doesn't have to be a particular, like a protein coding gene. There are snippets of RNA that are being transcribed all over the genome. Some of those can be miRNAs. Okay, so intragenic miRNAs, they can be found in the introns, they can be found in the exons of genes that code for proteins. Will intronic miRNAs, the ones found in introns, always be transcribed with the host gene? Give you 2 minutes to discuss. What are your thoughts on that? Okay. Can somebody tell me, will an intronic miRNA always be transcribed with the host gene? Anyone from the back of the class? Nah, you don't want to. Okay, go ahead.
So, in the case where the host gene is being transcribed, the input to the something exon to be transcribed from the premature mRNA, which then will be spliced out when the process the mRNA gets mature mRNA. So, when the host gene is being transcribed, it's the intronic mRNA.
Okay. When the host gene is transcribed, you are correct. The polymerase can't jump and avoid the intron. It just transcribes the whole gene. And if the miRNA is encoded by the intron, it's going to transcribe it with the gene, right? Now, yes, and there are situations, though, even though it's in the intron of the gene, it might also have its own regulatory elements in which it might be able to be transcribed when the mRNA is not transcribed. Does that make sense? So imagine a gene within a gene, that kind of idea. So it can also have its own regulatory sequences. Yeah. Okay. Let's do some clickers. Are host gene mRNA and target mRNA the same? So the host gene would be the gene that harbors the mRNA. So let's just say this would be the host gene here, this mRNA encoding gene. And is that always going to be the target? Five, 4, 3, 2, 1. Wow, well, almost a 50-50. 57% say B, usually no, and 43% say usually yes. So, okay. Why, I guess, why no, why yes? Why more people said no than yes? So why no? Who wants to volunteer a no answer? Yeah, don't be shy. Go ahead. Oh, that's so you're thinking about the downstream consequences, right? Like if it's always transcribed with the gene, it would always be silencing that gene. Yeah, for the most part, you are correct. But I'm like, imagine if dicer's not present, right? Or some of the enzymes that cut it up and are not there. There could be ways to regulate that, but so I understand what you mean. So as long as the target is going to be present, that mRNA, if it was transcribed with the host gene, it would be targeting that host gene. So for the most part, yeah, you're correct. But okay, so usually that is usually no. Usually it's not the whatever this gene that's transcribed. It's generally not the target gene. Okay, so and imagine like this is in the intron. So after splicing, that target that the mRNA it can be complementary to actually is no longer present in the transcript. So if you're really thinking about this, well, where does this all take place? Where does the silencing happen? Before the transcript, the intron is removed by splicing, which would mean it happens in the nucleus, or does it happen after splicing occurs? What happens after splicing happens? Where does the mRNA go after splicing? Okay. Don't tell them. Don't tell. Where is the mRNA having its effects? In the cytoplasm of the cell producing the mRNA, in the nucleus of the cell producing the mRNA, in the cytoplasm of the cell producing the mRNA, and neighboring cells. Where? Was this in the video? I don't think it was it? The where it all happens. Maybe it's in the video and the details of the video. Okay, 5, 4, 3, 2, 1. 57% say it happens in the cytoplasm. Great. Why do you say the cytoplasm? Why do you say the miRNA is having the effect in the cytoplasm? Yeah. Yeah. Actually, you were saying the original mechanism was to cut the the target RNA, right? Where would that one happen? Where could it happen? It could happen in the cytoplasm as well. Anywhere else you think it could happen? It could happen in the nucleus. Okay, if the mechanism of action is actually for risk to cut the RNA, target RNAs, that can happen anywhere the RNA is. It can happen in the nucleus, it can happen in the cytosol, it can happen outside the cell, it can happen in a neighboring cell, it can happen, we'll get to it, outside of the organism. Okay, we have immune systems. It can happen in all those places where the RNA is found, but if it does the inhibiting translation process, you know, when there's a mismatch between the miRNA and its target, it inhibits translation. That's what we said. Where does translation happen? This should be a unanimous screaming, yelling, we all know the answer. Where does translation happen? Cytosol. Okay. I imagined it happened much louder than that, but okay, good. Okay, so, yeah, that's the case. So Deisker, Risk, and everything, they can do their actions if they're inhibiting translation, they're going to do their actions where translation is found. They can also chew up RNAs in the nucleus. They can become nuclear localized, and they can go into the nucleus to cut up RNAs. They don't inhibit translation there because translation doesn't occur there, but anyways. Okay. In groups of 2 to 3, head to the activity worksheet in an E-class. I'm going to give you 10 minutes. Do the activity. We'll talk about the answers. It should be visible now. And as usual, there's a crowd mark hand in. Please remember to put all your partners' names on the hand in.
Recording stopped.
Okay, just a minute or so left. Okay, finish up and submit, or you can submit later. It's up to you, but gonna get back to it.
Recording in progress.
Okay. Okay. If the miRNA can be, if an miRNA can be exported from a cell, what can happen? What could it do? Yeah, go ahead. You had your hand up first. Mm-hmm. Very good. Very good. Yeah. So if an miRNA can be exported, and many of them are, we actually know this. For example, in plants, you got to remember I'm a plant researcher, so I get plant examples usually come first. But you find them in the xylem sap. So if you cut the xylem is the vasculature that translocates water and minerals and stuff upwards from the roots to the leaves. They are present in the xylem. They're translocated around the plants, some of them. Okay, so that's an example. Another example, I mean, we excrete them too. So what do they do? What do you think they do? So Christian was saying they can go to neighboring cells and they can, if they're imported, then what would they do? Don't be shy. Yo. I like it when you took her hand and you put it up last time. That was funny. Yeah. Oh, that's me. Yeah, they find the target mRNA, could be an mRNA, they find the target in that cell, right? It would inhibit translation in that cell. So it could be another cell produce the mRNA, goes into, exported out of that cell, taken up into a new cell. In the new cell, it inhibits the translation. It would do what the mRNA normal mechanism is to do it. It would inhibit translation, could result in destroying the transcript, having the transcript cut up into pieces. Depends on if the target is complementary, 100% complementary or not. Some plants secrete miRNAs that can be picked up by other organisms. For example, a fungus. What function might these miRNAs have? Five, 4, 3, 2, 1. Okay. 47% say A, to communicate with other organisms. 13% say to find mates. Yeah, plants are not really good at, if they secrete an mRNA, how would they find a mate? I don't know about that one. 40% say to exchange genes. Are they exchanging genes per se? Are they exchanging genes whenever they traffic out their microRNA and another organism takes it up? Exchanging genes. Does the microRNA get embedded in the genome and now it's a new gene for that organism? No. Okay. So they're using these microRNAs to communicate with other organisms. Can you talk to a fungus? I don't know how to talk to a fungus, but guess what? Plants can. Plants are sending messages to fungi. And they're changing the communities, the microbial communities that surround their roots. Okay, they can inhibit the growth. They can do various things. So some plants secrete mRNAs and deliver them to a fungus. What role could they play? So I kind of blabbed too much on that one. But what are some various roles that they could play? Yeah. Yes. Yep. Yep. So they can reduce a particular species that they don't that is threatening. Okay. So imagine they're inhibiting a gene in the fungus that's essential for the fungal growth. And remember, it's anyways. So they can inhibit the fungal growth. They can also actually promote the abundance of a particular fungus. They could promote the secretion of certain molecules from the fungus that the plant likes. Okay, they could potentially. Well, I'm not going to get into the mechanisms just yet because we talked about that. Applications. Why might this be better? Oh, so why? What are some applications we can use these microRNAs? So we could potentially make our microRNAs. Actually, they're very easy to make. I don't know how to make them. They're very easy to order. You can just order them from a company. Easy. Tell them the sequence you want. All the bases, you order it. Bing, shows up a few days later in a tube. Resuspend it. Spray it on plants. What would you be doing if you were spraying it on plants? What do you think could be useful application of microRNAs? Spraying them on plants. Yeah. Yeah. Yeah, brilliant. You're thinking ahead. Excellent. I like that. Good thoughts. So Christian was saying that fungicides, various pesticides are non-specific. Okay, they're just chemicals, and they could be they're non-specific in that they often target particularly conserved. You know, the manufacturers want to go for the big bang for their buck, so they want to try to kill as many fungi as possible, for example. So they're going to make a chemical that it targets a conserved enzyme, and it blocks that enzyme, the enzyme needed by the fungus to survive, by many species of fungus to survive. Kill all of them. Kill them. That's what the pesticide companies are doing. But that's really not good because maybe sometimes you actually want to be very specific, right? And so if you sprayed a microRNA and it was specific to a particular pathogen, it could be targeting that particular pathogen to arrest its growth. So then I did this. They're showing you spraying. People don't usually spray. I guess you probably could. Very expensive. They engineer plants, rather. They engineer plants to express. There's plants out in agriculture right now. May have consumed some today that actually express engineered microRNAs that'll provide resistance to nematodes, certain pathogens. How is a microRNA finding its target? just right down to the molecular level. That's it. Complementary base pairing, right? That's what determines what a target is if the microRNA can fit and it directly finds a very close match or an identical match, it's going to bind the target. Okay, so you can imagine the plant just like excreting or exuding all of the like vesicles that contain all kinds of contents, and they burst open and release these microRNAs into the rhizosphere, into the soil, and then any nearby fungus or bacteria or whatever. You've heard of multi-drug resistance in hospitals, how bacteria, they take up DNA from neighboring bacteria and they can acquire resistance. Well, they can also acquire microRNAs. And in the case of a fungus, eukaryotic cells, that can you can use that to your advantage to target essential genes to shut down their growth. Okay, so applications for double-stranded RNA, you can use them to target pathogens, but the concern is always whether you're going to actually accidentally target some other species that you didn't mean to. Okay, so we have a lot of sequence genomes for so many different species. So this can be tested on a computer before it even gets out into the field. But of course, you're going to want to do trials to make sure that these things are safe. So never fear, don't be scared. These are more accurate than the chemical pesticides that were used since before our parents were born. Yeah. Are they as effective? To tell you the truth, I haven't delved into the studies to know for sure, but I remember reading some early studies, and I remember reading they are highly effective, especially for nematodes, like trying to invade and go inside the plant, because as soon as they get into the plant, that they're right there. All the miRNAs are there. Yeah. You mean an miRNA? Difficult, very, very difficult. Because they would have to mutate enough so that the miRNA is no longer complementary. It would take quite a few changes in order for that to happen. Actually, it's less likely for that to happen usually than a chemical pesticide binding to an active site. Usually, maybe one or two changes could be enough that it doesn't bind. Yeah. I think I understand the direction you're going, but synonymous is, that's reserved for amino acid coding things, right? If the miRNA binds to the, if it doesn't match completely, it's going to inhibit its translation, right? If it's a perfect match, it'll cause cleavage. I don't, if you want, we can talk about it later. I don't know if I'm 100% getting it. Or if I answered your question or not. Yes. Yep. Mm-hmm. if the mRNA mutates to become 100% complementary, it's like even worse because it gets cut, it gets degraded, right? And that's more, that's a good point. So the cutting mechanism is more efficient, more effective than the inhibiting translation mechanism. When you inhibit translation, you bind to one mRNA and you're just blocking the translation of that mRNA. When in the cutting mechanism, if you're 100% complementary, that mRNA gets cut, quickly we go on to the next one, it gets cut, quickly we go on to the next one, it gets cut, and it just propagates on and on like that. I'm really curious, I didn't quite understand your question. So if you want to talk after class, or yeah, it'd be cool. I'm afraid I'm missing something. I want to get through all the slides there. Okay, so can plants communicate with each other? Everything else we're talking about a plant talking to a fungus. So chances are if a plant can talk to a fungus, it can probably talk to another plant. Okay, surprisingly, it turns out that plants can talk to each other via miRNAs. See, that's what I was talking about. It gets excreted from one plant and taken up with water, taken up into the other plant and moves through the xylem. It's going to be imported into the cells. This plant doesn't know if the miRNA came from itself or came from another plant. It has no idea. So as long as this plant here can exude it, can excrete it into the soil, and it's in the vicinity of another plant that can take it up, it can communicate. Why might it want to do that? Well, like I think I mentioned, well, maybe not in this class, but in plants all, they talk to each other through chemicals. They excrete gases that actually warn a neighboring plant, I'm being attacked, protect yourself, turn on your defense. There's ethylene and there's jasminates and there's other hormones that they warn each other. And they can also communicate to each other through miRNAs. Yes. To harm a neighboring plant, I would imagine, I would never say no. Has it been identified? I don't know. It absolutely could work. It absolutely, you can imagine if a plant was excreting an miRNA for which it did not have a target, but its competitor plant, like the allelochemical, has the target. Just like an allelochemical would work, one plant is kind of resistant, doesn't have the target. The other plant is not. Yeah, cool. I don't know. When you open your lab, please make that your first experiment to figure out. Okay, so you're studying a gene called attention, and you want to find out what the function of the gene is. How might you do this given what we've learned about this week? How might you think about this? I have a SNP, and nearby my SNP, I have a gene, and I think it's causing my phenotype, and now I want to figure out, what are you going to do? Yeah. RNAi interference, yeah. Very good. So you design an miRNA that's going to be complementary to that gene of interest, introduce it into the organism. And so we're going to talk about how that's done. You'd introduce it into the organism, and essentially the result would be that gene's expression would be reduced. Either the transcript gets cut up and it's not translated or doesn't do its job, or translation is inhibited if it's not a complete match. Yeah. Okay. So we can use RNA interference in the lab to study genes. We do this all the time. It's just a very, very rapid way to study gene function. We do it in cannabis. We do it in soybean. Okay, so here we go. So FOXP2 expression has been shown to be regulated by miRNAs. When miRNAs complementary to the coding sequence of FOXP2 mRNA are present, you would expect that the rate of FOXP2 mRNA degradation and the rate of FOXP2 protein production to what? So we got mRNA degradation and protein production. Which one's going to increase? Which one's going to decrease? Five, 4, 3, 2, 1. Okay, 64% are saying B, and then we have an equal dispersion of the other answers. So B, so you say that it's going to cause mRNA degradation to increase and the rate of FOXP protein production to decrease. B is the correct answer. Very good. Does everyone see that? Causes the degradation of the mRNA that increases. mRNA is getting chewed up, and it's thus decreasing the rate of the protein production, the rate of translation, because there's not as much target there to translate. Does miRNA change to mRNA or protein? Question mark. When are you measuring stuff? Yes. So you're saying, I would measure the amount of protein and I would expect it to be reduced. That's what you're saying? Good, good. And so the answer to the question is, does the miRNA change the mRNA or the protein? Does it change the protein? It does not change the protein. That's correct. It would just reduce the amount, its amount, right? Very good, very good. Yes. And does it change the mRNA? Yeah. Yes. Yeah. It's not gonna change the code or the nucleotide sequence, but it's gonna cause its degradation. So it's cleavage and degradation. Yeah, sure, if it's a direct match. If it's an indirect match, though, most mRNAs they got 1 or 2 nucleotides mismatched to their target. And so they're just gonna sit on that mRNA and they're not really gonna change anything, really, right? Okay, so, oh my goodness, 2 minutes left. I think I just have a few slides anyways. But anyways, we can use miRNAs to study gene function. You can take cells, you can take these huge multi-titer plates, 96-well plates we call them. Each well has a different miRNA, and you incubate cells. It could be a human cell line, cancer cell line, or whatever. Incubate cells in there, and they take up each well a different miRNA. So you're knocking down the expression of a different gene. So you can screen many, many, many genes with high throughput to try to figure out if they have a role in causing a particular phenotype. You can also micro-inject them into cells. We engineer them. We just engineer a hairpin DNA, like I was telling you. So when it's transcribed under a strong promoter, makes a hairpin, and then we have the gene silencing. Okay, so we use RNAi in genetics to study gene function all the time. It's very common. Causes RNA degradation. Okay, so that's just one level. You got to remember that genes are regulated at many other levels. It can be protein activation, translation, so on. And it's very complicated in that there can be gene networks. So like a transcription factor can turn on the expression of an miRNA. It can also inhibit the expression of a target, and that miRNA can inhibit the expression of a target. I'm not going to keep talking because you guys are all done. So, okay, see you later. See you next week. Thanks, guys. That's it. That's it. Yes.